While I'm introducing our speaker, grab your cookies and brownies and take a seat. But today it is my pleasure to introduce our speaker for the day, Dr. Kate Sales. Dr. Kate Sales just finished up his PhD here at U Calgary, so she's been kind to present her PhD work today with us. But just to give you a little bit of background on Kate, she did her Bachelor of Kinesiology, which she finished in 2015 from here at U Calgary, and then started a Master's of Kinesiology in Exercise Physiology, which was course-based, which she finished in 2019. From there, she started a PhD in Nutrition, Metabolism, and Genetics, supervised by Dr. Raylene Reimer. And as I mentioned, she defended that in 2025, just recently, and she was studying the link between exercise and the gut microbiota. When I asked her about a fun fact, she couldn't think of one, so I'm going to try and make up one here. Well, first of all, fun fact, I think she's the only one that is like the exact same height as me at the age ago, which is really nice. I don't have to strain my neck to talk to Kate, so we talk a lot. And then another fun fact, Kate is really, really helpful and sweet and is a very good friend of mine now, having been here for so long. And when I was making and building my house and designing it a couple years ago now, I wasn't sure if I should get matte white tiles for my backsplash, and at the same time she was renovating her kitchen, so she kindly offered to spill like sauce and curry on her white backsplash and test whether it comes off. So that just goes to say how nice Kate is and how helpful she is, and it did come off, and I do have a white backsplash, so if anyone was planning to do that. But today she will be talking about her PhD work, as I said, and she will be presenting her talk titled Exploring the Bidirectional Relationship Between Exercise and the Gut Microbiota, the Impact of Training Intensity and a Symbiotic Intervention. So please join me in welcoming Kate Sales.
Thanks, guys. Is this working?
Is it on?
Yeah. Yeah.
I can hear clicking.
Probably because I'm moving it around. Is that better?
Yeah.
Okay. Okay. Thank you everyone for coming to my presentation today. So yeah, like Ashna said, I'm going to present my PhD work today. Basically, this is the presentation that I did for my defense. And I basically just added some more introduction to it and expanded on my results a little bit. So today we'll go through some background on the gut microbiota and its link with exercise, and then we'll go through the results of my 3 PhD projects. So the gut microbiota is the collection of all microbes that inhabit the intestines, and this community is comprised mainly of bacteria. But it also includes other microbes such as viruses and fungi as well. It's now well known that the gut microbiota has significant influence on various physiological systems, including the gastrointestinal, the immune, and the nervous systems. We also know that the gut microbiota itself is a very dynamic environment that's influenced by a variety of mostly environmental factors, at least in the adult gut microbiota. The infant gut microbiota is a little bit different. But factors such as diet, antibiotic use, probiotics, prebiotics, these are all known to have either positive or negative influences on the gut microbiota. And for that reason, there is growing interest in trying to understand how we can optimize the gut microbiota through interventions that positively modulate either the structure or function of the gut with a downstream goal of improving health outcomes. So we know that the gut microbiota is influenced by these certain environmental factors, with diet probably being the most influential one. But what we've seen with more recent research is that exercise also seems to be a factor that influences the gut microbiota. So there have been a fair amount of cross-sectional studies that have shown associations between the gut microbiota and exercise outcomes, such as VO2 max. And there have also been a handful of exercise intervention studies that essentially look at how we can modulate the gut microbiota through different types of exercise interventions. And the large majority of these have been in aerobic-based exercise interventions. And then most recently, we've also started to see some research showing that in addition to exercise influencing the gut, that the gut microbiota can also influence exercise performance. And this study that was done by Jonathan Schiemann and this group out of Harvard in 2019 was really the first to show this relationship in humans. And this has become probably the most impactful study in the gut microbiota and exercise field. And what this group basically showed was that lactate produced by muscle fibers during exercise can enter the gut, where a very specific bacterial species is able to consume that lactate, which produces a beneficial microbial metabolite. As a byproduct, that metabolite can subsequently leave the gut and bind to receptors on skeletal muscle and modulate skeletal muscle metabolism. That's kind of their one proposed pathway for an ergogenic effect. And the other pathway is that by consuming lactate, they argue that the gut microbiota essentially acts as a metabolic sink for lactate, which also has performance implications. So they kind of propose these two ergogenic pathways. And they believe so strongly in their results that they actually went as far to isolate this bacterial species and develop it into this type of probiotic, which is now commercially available and is being marketed as this kind of fatigue fighting ergogenic aid for endurance athletes. So if you want to get yourself some Vianella, you can let us know how that goes. So we see this bidirectional relationship between exercise and the gut microbiota, whereby exercise training, whether it's acute or chronic, influences the gut microbiota, and the gut microbiota can also influence exercise performance. So I'll get into this a little bit more in the next couple slides. But before we do that, I just want to briefly cover how we actually analyze and assess the gut microbiota. So anytime we do any gut microbiota analysis, we collect the stool sample. And our first step is to analyze that. And there are kind of two main analytical techniques that we can use. The first is called 16S rRNA sequencing. So all bacteria have the 16S gene, and that 16S gene has highly conserved regions, which are the same among all bacteria. And then it also has hypervariable regions, which differ between bacteria. So what we do is we select one of these gene regions, and we amplify that region using PCR. And then through sequencing techniques, that generates thousands of reads, which are just DNA sequences. And then through various bioinformatics pipelines, we can actually identify what bacteria are present and their relative abundance. So 16S sequencing, again, because we are using that 16S gene that all bacteria have, that limits us just to looking at the bacteria. So we don't get any assessment of any information on the other microbes in the gut as well. And it essentially tells us who is there. So it's a taxonomic assessment of the gut microbiota. The other one is called shotgun metagenomics. So with shotgun metagenomics, we are sequencing all of the DNA in the sample. And that, through our sequencing technique, generates millions of reads. And then again, through these bioinformatics pipelines, we can identify which microbes are present. And now we're not only limited to bacteria because we're sequencing all of the DNA of the microbes. We get their relative abundance, and we also are able to identify what genes and pathways they carry. So again, we get kind of a more robust assessment of the gut microbiota because we get bacteria, but we also get fungi, viruses, etc., all the microbes that are present. And the real benefit of shotgun metagenomics over 16S sequencing is we get to see who is there, but we also get to see what they're doing. So we get that functional profile as well. The other benefit of shotgun metagenomics over 16S sequencing is that it has a higher taxonomic resolution. So with 16S sequencing, we can often get down to the genus level, but with shotgun metagenomics, again, because we're sequencing all of the DNA, we can resolve down to the species level. So it's just a much more sensitive and robust approach to analyzing the gut microbiota, but it comes at quite a significant cost. And you need very skilled bioinformaticians who are able to work with these kind of data sets. So for all of my analysis, we use 16S sequencing, but our lab does also do a little bit of shotgun metagenomics when the money is available. Okay, so once we've sequenced the gut microbiota, we get that raw data, again, which are just these DNA sequences. And then through those bioinformatics pipelines, we transform that data into usable information. And there's a few common metrics that you will see in all of gut microbiota research that essentially are used to assess the gut microbiota. The first one is bacterial diversity. So we have alpha diversity and beta diversity. With alpha diversity, we're looking at the diversity within one sample, so the diversity within one environment. And alpha diversity is characterized by the richness, so that's the number of unique bacteria that are present, and evenness, so their distribution. And typically, what we see with a healthy gut microbiota is that it's characterized by high richness and high evenness. So that would kind of be like your gold standard for your gut microbiota. Alpha diversity. The other one is beta diversity. So with beta diversity, we're looking at the diversity between two samples or communities. So the diversity between environments, and beta diversity describes how similar or dissimilar two communities are from each other. So with beta diversity, these are usually visualized on a PCOA plot. We're looking for clustering. So if we have two communities and we see that they're clustering very closely together, we can say that they're quite similar, but if we see distinct clustering, then we can say they're quite different. So with the beta diversity, we're actually calculating a distance metric between the communities to, again, look at the similarity or dissimilarity. The other thing we look at is the relative abundance. So this looks at the abundance of a single bacteria at any taxonomic rank in regard to how much of the total community it makes up, and that is represented as a percentage out of 100. Differential abundance, so this is specific for when you're comparing two conditions, so that could be two groups, 2 time points, 2 treatments, whatever, and you want to know which bacteria are significantly more or less abundant in one condition versus the other. And then lastly, we also look often at fecal short-chain fatty acid concentration. So short-chain fatty acids are microbial metabolites that are produced when the gut microbiota ferment fiber or other specific components. And these are really important for maintaining the gut barrier, and they can also leave the gut and act on peripheral tissues as well. So we will also often look at fecal short-chain fatty acid concentration. Typically, you want a high fecal short-chain fatty acid concentration. Okay, so back into this bidirectional relationship between exercise and the gut. So what we see from cross-sectional studies is that athletes and individuals who are more aerobically fit tend to have more favorable gut microbiota profiles compared to sedentary controls. And their gut microbiotas are usually characterized by high microbial diversity and enrichment of short-chain fatty acid producing bacteria. Exercise intervention studies, these often demonstrate that exercise training can positively shift the gut microbiota by increasing microbial diversity as well as the abundance of health-associated microbes. And again, the exercise intervention studies are largely done in aerobic training interventions. We don't really see much research with resistance training. And supplement intervention studies and other mechanistic studies, these show that certain bacteria and their metabolites, which we have just discussed, can influence exercise performance through mechanisms that either impact lactate clearance or augment skeletal muscle metabolism. So there have been a decent amount of studies that have explored this bidirectional relationship, but we still have a lot of gaps in the literature. So exercise intervention studies, these are largely these are arguably the most robust approach for understanding how exercise influences the gut. But we still really don't know the role of intensity in kind of modulating these shifts. Many studies that we have right now, they've made conclusions regarding the role of intensity, but the large majority have used exercise prescription approaches that we now know don't accurately control the exercise stimulus. And then they've also used single arm design. So you really can't make these direct comparisons with other intensities. Cross-sectional studies in athlete populations have provided some good initial insights into the effects of years of chronic sport-specific training on the gut microbiota. But they've been conducted mainly within a single sport and have kind of failed to integrate other training variables that we know influence the gut microbiota, such as training volume. And lastly, supplement intervention studies, which are arguably our best approach for understanding how the gut microbiota influences exercise. Almost all have used probiotic interventions, and we really see variable findings with probiotics in regards to their ergogenic effects. Now, this could be due to differences in the intervention duration, the bacterial strain use, the dosing. But it could also be that probiotics themselves are just not a strong enough signal to modulate the gut microbiota in a way that will lead to downstream improvements in exercise performance. So that brings me to the purpose and aims of my thesis. The first was to explore if exercise intensity differentially influences the gut microbiota. So for this project, we recruited 65 participants who were a subset of participants from a larger exercise training intervention study who had provided essentially just additional consent to participate in our sub-study. So in addition to all of the exercise training that they were already doing as part of their exercise intervention study, they also consented to providing a stool sample, a 3-day food log, and a blood sample. And after baseline exercise testing, they were randomized to one of 5 work-matched exercise groups according to exercise intensity domains or a non-intervention control group. So all participants except the control group completed 3 exercise sessions per week at their prescribed intensity for 6 weeks. And after the 6-week period, everybody returned, including the control group, to complete the exact same testing measures they did at baseline. And for our analysis, in order to increase statistical power, we ended up combining sit and hit into severe, heavy 1 and heavy 2 into heavy, and then we left moderate as is. So we could analyze effects basically at the exercise intensity domain level instead of the individual exercise training group level. The first thing we wanted to do was to see if 6 weeks of aerobic exercise in general increased alpha diversity. So what we did is we pooled all exercise intervention participants into just an exercise group, and we looked at changes in alpha diversity from pre to post, in which we saw an increase in alpha diversity in the exercise group, but not in the control group, suggesting that 6 weeks of aerobic exercise does shift the gut microbiota. Next, we wanted to see if these changes were specific to intensity, and we saw that heavy and severe, but not moderate intensity exercise significantly increased alpha diversity, and this suggested to us that there is this kind of minimum exercise intensity required in order to induce shifts in the gut microbiota. We then looked at beta diversity at post testing to see if we could observe any clear clustering of any of these exercise groups, but we didn't see any significant differences. So these findings indicate that 6 weeks of aerobic exercise shifts the gut microbiota in an intensity dependent manner. So we already had answered our primary question about the role of intensity. And next we wanted to dive a little bit deeper and see if independent of intensity, if there are distinct gut microbiota profiles in participants who responded better or worse to the intervention based on a post-intervention improvement in VO2 max. So we recategorized participants as either a responder or non-responder based on a post-intervention increase in VO2 max by greater than or equal to 3.5 mils per kg per minute. And we ended up with 25 non-responders and 32 responders. When we looked at alpha diversity between these groups, we saw a significant increase in both the non-responders and responders. And when we looked at beta diversity, we again saw no differences, no clear clustering. So diversity and differential abundance analyses, which we did do differential abundance, but I didn't show it here just because it didn't show too much. They're very good at highlighting specific microbial features that are associated with an outcome, our outcome being exercise response. But they are quite a granular analysis approach, and they don't really capture the bigger picture in what is a very complex environment. So still looking at responders and non-responders, we next use a random forest classification model to extract bacterial features that were considered important for distinguishing responders from non-responders. And one I just want to point out here, because it will continue to come up, that was considered important for non-response was Holdermanella. Next, we performed a balance analysis, which essentially finds 2 groups of bacteria that when you compare those 2 groups of bacteria as a ratio, best separate responders from non-responders. And what we see here is that in the non-responder group, in the gray here, they align a lot more with the group B bacteria, whereas the responders in purple, they're kind of smack dab right in the middle. And again, we see here in the non-responders, Holder-Manella coming up. We then wanted to see whether this balance of bacteria mediated our outcome of exercise response. And we saw that it did in the severe group, but not the moderate or heavy group, suggesting that the balance of bacteria is especially important for exercise response with severe intensity exercise training. So these findings indicate that Holder-Manella is a feature of non-response and that the individual taxa as well as the community balance matter for exercise response. So knowing that certain taxa and the broader microbial community are important for response, we next used a fast and frugal decision tree to see if we could essentially predict responder status based on the presence and abundance of bacteria at pre-testing. And this once again extracted Holder-Manella as the most important bacteria in predicting non-response. We then decided to dive into Holder-Manella a little bit more. Clearly, this is very important for exercise non-response at least. And what we found was that Holder-Manella abundance was much higher in non-responders at both pre and post. And it's basically almost absent in responders at the same time points. But when we looked at its change from pre to post, we really don't see any change. So this suggested to us that it's the presence of Holder-Manella rather than its change over time that seems to be important in non-response. We then used multiomics to integrate our three data sets, being the nutrition, serum, and microbiota. And this once again extracted Holder-Manella as a feature important in non-response. And it also revealed several inflammatory markers as features of non-response. This is both serum and gut inflammation. And for gut inflammation, this included zonulin, which is an intestinal permeability marker. And then when we took our blood samples and we looked at serum analyte concentration, we also saw significantly higher serum zonulin concentration at post in the non-responders. So these findings indicate a co-occurrence of Holder-Manella and zonulin in non-responders that may relate to gut inflammation. Okay, so the last thing we did for this study was a correlation network using the diet and microbiota outcomes to see if we could observe any links between or within these data sets. And this network is at post. It only shows correlations that had an R value of 0.7 or larger, which are shown by the green lines. And the thicker the line, the stronger the correlation. So on the left here, we have the responders. On the right, we have the non-responders. And something that was really interesting that came up here was in the responder group, we see Holder-Manella come up, but we don't see Holder-Manella in the non-responder group. And that would seem contrary to our results up until this point. But if you look at Holder-Manella in the responder group and you see the other bacterial groups that it's correlated with, many of these are very beneficial bacteria. And within the non-responder group, because we don't see Holder-Manella come up, that means it doesn't have strong correlations with other bacterial groups. So this led us to think that in responders, in people who are responders who have Holder-Manella, Holder-Manella exists within a compensatory niche, whereby you have other beneficial bacterial groups that are basically diluting the negative effects of whatever Holder-Manella is doing. But in the non-responders, Holder-Manella, we know that they have it from our previous analysis. We know that it's in high abundance, but it exists in this isolated niche. So basically, this isolated niche can allow Holder-Manella, whatever negative effects it's having, to essentially have a larger effect. So this led us to conclude that there's kind of two pathways for being an exercise responder, regardless of exercise intensity. You either don't have Holder-Manella, or you have Holder-Manella, but it exists in a compensatory niche. Okay, on to my next project. So our second aim was to explore gut microbiota profiles in competitive athletes across broad sport categories with a within and between category focus on training volume. So for this study, we recruited 99 current competitive athletes from any sport, ranging from local all the way up to Olympic level athletes. And at a single time point, we collected a stool sample, a 3-day food log, a GI questionnaire, and a sport and training questionnaire. And based on their responses to the training questionnaire, we then subsequently categorize athletes into one of three broad training categories, being endurance, mixed, or sprint power. So our first step was to look at what factors within our data set explain variability in the gut microbiota community structure. And we saw that sport category alone was not a significant factor, but when sport category was combined with weekly training hours, which we refer to broadly as training volume and defined as training less than 15 hours a week or more than 15 hours a week, we see that this two-factor relationship explained the most variability in the gut microbiota community structure within our data set. So based on this, we focused on our subsequent analysis on this combined relationship between training category and training volume. So we first wanted to see how training volume influenced enrichment and depletion patterns of bacteria within each training category and essentially how they overlap with other training categories. So to do this, we used a Venn diagram to visualize the number of unique and shared differences of bacteria by training volume within each training category. And we use this in conjunction with a heat map, which I've showed an example of here. You don't have to worry about it. I just wanted to kind of show you how we use them together. But basically the Venn diagram tells us how many bacteria are differentially abundant in these comparisons. And then we can look to the heat map which actually shows us which bacteria and in what groups they're enriched in. But just focusing on the Venn diagram for now and the number of differentially abundant bacteria, we saw that in the endurance category, 63 unique bacteria differed according to training volume, followed by 108 in mixed and 120 in sprint power, suggesting that the gut microbiota of endurance athletes is the least sensitive to training volume because they have the lowest number of differentially abundant bacteria between the two training volumes. Whereas the gut microbiota of sprint power athletes is the most sensitive because it has the highest number of differentially abundant bacteria between the training volumes. So these findings indicate that the gut microbiota profiles differ within training category according to training volume. For our next analysis, we wanted to examine differences between training categories at each training volume to see whether bacterial differences essentially appeared or disappeared according to training volume. And when we stratified by training volume and we performed pairwise comparisons between training categories, we again observed these unique and shared bacterial differences between training categories. So at both training volumes, so less than 15 hours a week and more than 15 hours a week, the number of differentially abundant bacteria that were unique to one pairwise comparison between training categories was consistently higher at less than 15 hours a week compared to more than 15 hours a week, suggesting that basically at higher training volumes, the gut microbiota becomes more homogenized. So there are less differences between training categories. And at lower training volumes, the gut microbiota is more training category specific. So we see more differences between training categories. Further, at less than 15 hours a week, we see that there are three shared bacterial differences among all of these three pairwise comparisons. But essentially, once training volume crossed over that 15-hour-a-week period, these differences disappeared. And you can see that here in the heat map. At greater than 15 hours, we no longer have the number 3 there. And then when we look at the heat map, we see that these differences that did exist at less than 15 hours a week are no longer there. So again, just supporting the fact that higher training volumes homogenize the gut microbiota across athletes, across sport categories. So again, that's what these findings indicate. Higher training volumes homogenize the gut microbiota. Lastly, we wanted to look at some nutrition data. So we wanted to see specifically how athlete nutrient intake differs by training category and volume. So we performed a redundancy analysis, and this showed that training category and volume, both independently and collectively, explain variation in dietary patterns. And we can see the blue here, which is the endurance group. We see them kind of diverging away from the mixed and sprint power groups. So endurance athletes tend to have this kind of distinct dietary pattern, whereas sprint power and mixed athletes seem to have more similar dietary patterns. So for my final aim, we wanted to explore whether 6 weeks of supplementation with a novel symbiotic could improve endurance exercise performance using a randomized double-blind placebo-controlled parallel trial. So this was a MyTac supported study. We collaborated with a probiotic manufacturing company out of Montreal called Lalaman Health Solutions. They mainly focus on health outcomes, but they were also interested in the utility of their products for sport performance. So we essentially collaborated with them to come up with a novel symbiotic formulation based on bacterial strains and prebiotic fibers that they had available to test in an athlete population to see if there could be some ergogenic effects of probiotics and prebiotics. So we were kind of trying to, in a way, replicate the study that was done by the Harvard group, but by using an actual exogenous intervention with prebiotics and probiotics to see if we could replicate some ergogenic effects in a kind of endurance exercise model. So for this study, we recruited 21 trained cyclists who provided a stool sample, serum blood sample, and they also completed a GI questionnaire and 3-day food log to assess gut microbiota outcomes. And then they did some exercise testing as well. So they did a step round step test, a 16-minute ride in the heavy intensity domain, followed by time to exhaustion in the severe intensity domain, and a 20-kilometer time trial to assess, again, performance outcomes. And we also standardized diet in the 24 hours before the 60-minute ride in the 20-kilometer time trial. After they had completed all of their pre-testing, participants were randomized to either the symbiotic group, which was 15 billion colony-forming units of probiotics and 100 grams of prebiotic fiber, or the placebo group, which was 2 grams of potato starch for 6 weeks. And after 4 weeks, they returned for post-testing and they completed the identical measures that they did at pre. So our primary outcome for this study was the 20-kilometer time trial. And then our secondary outcomes were the gut microbiota related outcomes. So fecal short chain fatty acid concentration, serum analyte concentration, and the GI symptoms. So given that we hypothesized that our symbiotic would increase short chain fatty acid production in the gut, and that some previous research has shown that short chain fatty acids can modulate skeletal muscle metabolism, we had participants complete 60 minutes of heavy intensity cycling to assess substrate oxidation during the trial. However, we did not see any effects of the symbiotic supplementation on fat or carbohydrate oxidation at any time point during the trial, nor did it influence any other physiological measures, including average VO2, RER, heart rate, or blood lactate concentration. Similarly, for the 20-kilometer time trial, we also did not observe any changes or differences in time to complete the 20 kilometers. However, we did observe a numerically lower blood lactate concentration in the symbiotic group at 5, 10, and 15 kilometers throughout the trial, but it was not statistically significant. For our gut microbiota related outcomes, overall, we really didn't see much at all. Between the gut microbiota, the GI symptoms, the serum analytes, and the fecal short chain fatty acid concentration, really the only thing that we saw was an enrichment of this bacterial genus Lactobacillus in the symbiotic group at post relative to pre, and in the symbiotic group at post relative to placebo at post. And again, no other differences between the symbiotic and placebo group for those other outcomes. We also did something called strain recovery. So this was kind of a unique aspect to our study. Most studies don't do this. It's essentially a way to assess compliance and strain survival. So basically, we perform the strain recovery on our supplemented strains using PCR. And we saw that Bifidobacterium longum was recovered in 100% of the symbiotic participants in higher absolute abundances, while the Lactobacillus plantarum was only recovered in 67% of participants in lower absolute abundances. So key takeaways from this study are that the symbiotic did not influence substrate utilization during heavy intensity cycling. It did not decrease time to complete the 20-kilometer time trial. We did observe a numerically lower blood lactate concentration at those steady state time points during the 20-kilometer trial, but it was not statistically significant. In terms of our gut microbiota and serum outcomes, we did not observe any increases in short chain fatty acid concentration, nor did we see any influence on any of the serum cytokine concentrations. It did not substantially modulate the gut microbiota. We did observe that enrichment of lactoplanty bacillus, and we did have successful strain recovery. So to be honest, a largely null study, which was disappointing, but that happens sometimes. Okay, so in summary, my thesis shows that exercise intensity differentially influences the gut microbiota with a minimum intensity required in order to induce shifts. And we further demonstrated for the first time distinct gut microbiota profiles between responders and non-responders. We also showed that training category and training volume shape the gut microbiota profile in competitive athletes, within category analysis showing that sprint power athletes have gut microbiotas that are the most sensitive to training volume, and between category analysis showing that high training volumes appear to homogenize the gut microbiota. And lastly, we show that supplementation with our novel symbiotic formulation does not significantly improve endurance performance outcomes. So with that, I'd just like to thank my supervisor, committee members, examiners, and thank you all for listening, and happy to take any questions.
With that, the floor is open for questions. Who would like to go ahead? Lots of hands. Yeah, I'm gonna go in order. So question here, then Bill, then Jim, and then Erin. Remember that. Yes, go ahead.
Thanks for that, Kate. So I want to go back to the Holder-Manella.
Yeah.
So we know that it is a differentiating factor for responders and non-responders. In your readings, do you know, are there any other effects, like beneficial or negative, on health outcomes or any other aspects?
Yeah, so it's a unique bacteria. There hasn't been a lot of research that's been done on Holder-Manella. It's one of those bacteria that seems to be related both positively and negatively to health outcomes. So some research has shown that Holder-Manella is positively associated with a diet that's high in fiber, which would be, a good association. But then we also see data showing that Holder-Manella has been linked to a lot of inflammatory conditions. So it's probably, like, from an ecological perspective, it's probably context dependent. Like, one thing that when we submitted the paper, one of the reviewers said, which I agree with, it's never going to be one bacteria that's going to lead you to an outcome. But it could be that whatever niche that bacteria operates in, whatever other conditions might be present, that it's the combination of those things that could kind of lead to some downstream health, or in our case, performance effects. But it is like a short chain fatty acid producing bacteria. It also ferments protein, which is not a good thing. So it might kind of tailor its function depending on the person. Yeah.
Okay, Bill was next.
I just have a question about your first study. So you saw a difference only in the very intense exercise back there.
We saw, in terms of alpha diversity, we saw increases in both heavy and severe intensity.
I'm just curious, how long do those changes last? Do the athletes need to sustain that heavy intensity in order to maintain those?
Yeah, it's a good question. So basically, any changes that we see in the gut microbiota through any of these interventions are transient. And so it's kind of the downfall, I would say, of gut microbiota research is that whether you're taking a probiotic, whether it's a fecal transplant, exercise intervention, as long as you are on that, you seem to have the whatever beneficial effect or negative effect of its antibiotics, for example. But as soon as you stop that treatment, you will revert back to your previous gut microbiota profile. The gut microbiota is weird because it's very dynamic, yet remarkably stable at the same time. You essentially develop your gut microbiota in your first 3 years of life, And what you have at age 3 is essentially what you have until adulthood. And then we often see reductions in diversity into kind of elderly age. So, yeah, to answer your question, it's whatever effects you see, you usually have to maintain that exposure in order to continue with whatever outcomes you see.
Okay, and Jim?
I also have a question about that chapter, because that was it's like a test pretest.
Yeah.
Intervention, right?
Yeah.
Would it be worth adding, like rerunning the analysis, but some of the other studies data where you have like a sedentary population and a heavy endurance population to see whether before they have the fitness intervention, they're more overlapping with sedentary, and then like watch the transition towards endurance, because like endurance may have like elevated diversity during that transition phase where they have both the microbiota of a fitness one as they're transitioning to it, but still the remnants of like a sedentary community or composition.
Yeah.
And like maybe through that transition, the diversity is actually artificially higher as it's actually opening during the transition phase. Like, do they actually like lower down as they encroach into the full endurance, or is it like too idiosyncratic to make that comparison?
I don't know, like I don't think it happens linearly, like it's so like inter-individual. And it's hard because again, like it's when we take a stool sample, it's like one point in time. And diet, for example, you can see changes in the gut microbiota within 24 hours. And so it's also kind of an issue with gut microbiota research is that you're often sampling at one point in time and making inferences based on what you see then. But I think, and like what we also saw too in our study is that the changes are quite minor. Like the beta diversity plot is essentially it's capturing the overall community composition, whereas alpha diversity is kind of more granular. So you often will see in studies like ours that showed minor changes in the gut microbiota, changes in alpha diversity, but no changes in beta diversity. Because the beta diversity would be like large scale community restructuring. So yeah, there's probably like more sensitive like analysis techniques that you can use to detect more kind of like temporal changes. But yeah.
Aurelia?
Oh, yeah. Oh, very good presentation. Thanks. Congrats.
Thank you.
The first part of my question is kind of related to Leah. So this bacteria is what's named Salmonella.
Yeah.
You're kind of posing that it can act as an opportunistic bacteria, and it can affect permeability type junctions. And then, so is there any report that, like related to IBD, IBS, or Crohn's? And if so, is there any mechanism?
Yeah, not with Salmonella. I would say like our study is the first study to show a co-occurrence of Salmonella and gut inflammation. Even then, like we used like a ELISA kit for zonulin, which has inherent issues as well. But yeah, it's kind of a niche bacteria. We also didn't do any GI symptoms questionnaire in that group. So that would be another thing that we could kind of tack on in a future study. Like in a perfect world, you would isolate Salmonella, you would probably get a group of germ-free mice, you would, inoculate Salmonella in one and not in the other, and see if there's performance detriments in the Salmonella group. Yeah, like everything's very correlational, observational, not really mechanistic. Our conclusion about gut inflammation in Salmonella is simply our observation that high zonulin and Salmonella co-occur in that group, and that there is an increased serum concentration of zonulin in the non-responders. So we're just kind of taking these data sets. I mean, like, okay, what do we see? But we're lacking the mechanistic approach there.
I have a question.
Yeah.
So the changes that you saw or the differences you saw between like your training volumes, your different groups and categories, what in the order of magnitude, like how do they compare to changes in gut microbiota that you would see with like diet
Oh,
interventions?
Yeah, like the diet for sure would have the most robust and most kind of largest influence on the gut microbiota. And it's extreme diets, like our Western diets that are low in fiber, high in fat, perturb the gut microbiota a lot. Diets high in fiber are very good for the gut microbiota, specifically because the fiber in the diet is used by the gut microbiota for fermentation, which produces those short chain fatty acids, and that nourishes the gut barrier and is good for survival of other bacteria. So again, it's like it's difficult with human research when we're trying to isolate the effects of whether it's intensity, training category, because we have so many other factors at play. And that's why even in like our Permanova, if you look at the effect size, they're always really small in gut microbiota research, just because it's such a complex environment. And there's many factors that we can't control for. And so we see very small effects of these things on the gut microbiota. It doesn't mean that they're not meaningful or significant. It just means that, we're trying to shift a community that, with kind of a minor intervention often.
And you had food logs, so did you see
Yeah.
any like correlations or any sort of relationships between what people were eating and their results, or?
Just, we haven't done any like correlation network with the gut microbiota. We, in our first study with the Holdemanella, we did see positive correlations of Holdemanella with vitamin B12, which is a vitamin that comes from meat products. So, there often is associations there with what you're eating and how that influences your microbial community. But in our other studies, we haven't done any kind of correlations with those yet. Yeah.
Brent, and then Venus.
I don't know anything about the microbiome. But from my very naive viewpoint, it seems like it's really sensitive and you can always find things cross-sectional.
Yeah.
It's different in people with OA that don't in no OA. It's different in high, given as an intervention, prebiotic, probiotic.
Mm-hmm.
It just seems to me like there's lots of null results, like the interventions are not really doing.
Yeah.
Is that a fair statement, or are there actually like some really strong known applications of prebiotic, probiotic?
Yeah, it's a good question, because I think for us, we are kind of limited by the population who we recruit. Like, we see more robust effects in people who have an existing perturbed gut microbiota. So after, let's say, antibiotic exposure for a long period of time, you've essentially depleted the gut microbiota, and you can kind of effectively repopulate with probiotics and prebiotics. Same with people who have IBS or some sort of gastrointestinal conditions. They are more responsive to exogenous interventions. Within our group of trained athletes, we know that they already have, high microbial diversity. They have kind of an enrichment of beneficial bacteria. And so what we often see in healthy populations is that 50% of people are kind of resistant to colonization. And that's simply because you just don't have maybe a microbial niche that allows, like, from an ecological perspective, it's an invasion. Like, your gut bacteria will see anything coming in as an invasion, and it doesn't want it to populate, which kind of has good and bad effects. So that being said, though, you don't have to see, like, for example, an enrichment of bacteria or an increase in short chain fatty acid concentration in order for there to be an effect. So they can still be metabolically active and be having a functional effect without us observing it in our kind of proxy measures in terms of, you know, abundance and whatnot. So yeah, it's kind of I think like I was disappointed but not surprised, I think, with our results.
Well, yeah, are there examples outside of gastrointestinal disorders and things like that where you give it as an intervention, people get smarter or like
Yeah.
What pick your whatever.
Yeah, exactly. And like there are like I think there's a lot of people who are using like fecal microbiota transplants, other therapies for looking at mental health outcomes, for example. So it depends what outcome you're measuring. I don't think like a probiotic is not a strong signal, I would say. We also dosed quite low in the other exercise intervention or the other supplement intervention studies that we've done that I've seen. They're dosing at like 90 billion CFU a day. We're at 15 billion. So I do think that in some cases there's a dose response relationship, and potentially we were just too low to find something. We were also underpowered, and we have a population who already has quite a good microbiota. So again, we can't say that it's doing nothing because we don't know, but just from our measures, it's difficult to draw any conclusions so we don't see it in our analysis.
Venus, I think you had a question about that. I had a question about the mechanism that you about conditioning exercise and
Yeah.
which is basically lactic acid. So could you try to correlate between the different types of exercise and lactic acid and the changes things could be might happen?
From the study that had from before that the other group had done. So they so basically this was like a human intervention study. Actually, it wasn't even an intervention. They took people who were completing the Boston Marathon. They took a stool sample 5 days before, every day, 5 days before, and then 5 days after. And they looked at the gut microbiota to see which bacteria were enriched or depleted. And they found that this Vianella, was the only one that showed a significant enrichment after the marathon. And so they isolated that, and then they used animal models, and they inoculated mice with the Vianella, and they took them through different exercise interventions, and they saw that compared to a control bacteria, but with the same bacterial load, so a bacteria that doesn't catabolize lactate, that there were performance improvements in the Vianella group. And then by that, they basically hypothesized that when lactate that is produced by muscle fibers goes into systemic circulation, it can enter into the gut, and that Vianella is able to catabolize the lactate, and essentially acting as like a metabolic sink. So it's like an additional site for lactate oxidation. And that when it does that, it produces short chain fatty acid. And that short chain fatty acid can then leave the gut, enter back into systemic circulation, bind to short chain fatty acid receptors on skeletal muscle, and act as both a signaling molecule and additional substrate. So it sounds really cool, but there's a lot of issues with, I think, their conclusions. And I think the biggest one is that during high intensity exercise, like these are Boston marathoners, they're running, quite close to, say, anaerobic threshold. There's such a large redistribution of blood away from the gut towards the muscle. So I don't know their first part of their pathway of lactate actually making it to the gut. I think that's like the biggest issue, because you're redistributing blood away from the gut. So I don't know how much lactate is actually ending up in the gut. So it's very cool, but I don't know if it's legit.
Like took blood sample. So did you look at the lactate in the blood and try to correlate with what type of bacteria you have and the changes depending on the amount of.
In our last study, we just looked at, blood lactate concentration during the 20-kilometer time trial and some other ones, but we didn't also look at that kind of correlated with other bacteria, but that could be interesting.
Marika has a question.
Yeah, thanks for the presentation. Yeah, that was really great work. I have two questions. One kind of sparked my prior comment on what are your thoughts on like personalized or tailored gut microbiota interventions? I know like you did some great like modeling and classification work. I'm understanding that individuals have their own personal blueprint. And I'm wondering like whether you could do modeling or look at the introduction of maybe new strains or species and then see whether that would classify or change the classification for an individual?
Yeah, I think like with most things that it's the kind of individualized approach is the only way we can go from here. Everybody is like, they'll say that your gut microbiota is as unique as a fingerprint. And so there most definitely isn't a one size fits all. When it comes to the gut microbiota, I know that some companies who will charge you, $500 to send your stool sample in, and they'll do their own analysis, and they'll kind of tailor nutritional needs based on that. I don't think that we're there quite yet. I think it's like more complicated than that. But I do think that eventually we will kind of get closer to personalized gut microbiota approaches, whether it's for exercise, nutrition, health, whatever it is, but I don't think we're there yet.
My second question is about your second study.
Yeah.
If I'm understanding correctly, as the training volume increased, even across the high intensity, like the sprint athletes, for example, who did have higher alpha diversity, it kind of homogenized later on. So there was decreases in maybe the diverse strains. Do you think that could be because of like stress, training load? Like what factors could play into
Yeah, so that the diversity of the gut microbiota with the higher training loads didn't change. It's more so that we see less category specific differences at the higher training volume. So the like endurance versus mixed versus sprint power, they all seem to look more similar. So there's something either about that group of people, maybe athletes who are training more than 15 hours a week have more similar lifestyles, more similar diets. Or maybe it's just that training volume does have that large of an effect on the gut microbiota. That's what we like tend to see at those greater than 15 hours a week. And I forget the last part of your question.
that was it? Yeah.
Okay.
Any other questions for Kate?
In your study, Kate, how did you come up with your 15-hour week threshold?
Magic? No, so originally we had 6 different categories for training volume. But we were underpowered to look at all 6 of them. And so when we looked at the data, we kind of had a nice fairly even distribution between less than 15 hours a week and more than 15 hours a week. And there had been one other study that was in cyclists, and they saw that at that threshold, more than 15 hours a week, that there were different gut microbiota profiles in people at higher training volumes versus lower training volumes, independent of athlete status. So that was the main reason, but it was
Okay, well, if there are no more questions, then let's thank you for talking.